Hello everyone, I'm Jayden Hong,
And I'm Zayden.
and today we'll be presenting our biotechnology project. Our lab focused on human genetic variation, specifically investigating the TAS2R38 gene. This gene codes for a taste receptor that allows us to perceive bitterness. In our case study, we explored why some people find substances like PTC or certain vegetables incredibly bitter, while others taste nothing at all. Using DNA extraction and PCR amplification, we move from physical taste testing to a molecular analysis of our own genotypes. In Unit 1, we learned that the structure of macromolecules is held together by specific chemical bonds. In our lab, the double-stranded DNA molecule is stabilized by hydrogen bonds between complementary base pairs. This chemistry is critical during the PCR process. During the denaturation phase, we raise the temperature to 94 degrees Celsius to physically disrupt those hydrogen bonds, allowing the DNA strands to separate so they can be copied. We also rely on the polarity of the DNA backbone to move the samples through the gel later on. Moving to Unit 2, we focus on cell compartmentalization. Our target DNA is tucked away inside the nucleus, protected by the phospholipid bilayer of the nuclear envelope and the plasma membrane. To get our sample, we had to perform cell lysis. By using a specialized extraction buffer and heat, we disrupted these membrane structures to release the genomic DNA. We then use a centrifuge to separate the dense cellular debris, like organelles and proteins, from the soluble DNA supernatant we needed for amplification. Unit 3 is all about enzymes, which were the workhorses of this lab. We used two key enzymes, Taq polymerase and Hy3. Taq polymerase is unique because it is thermophilic, meaning it doesn't denature at high temperatures, allowing it to catalyze the synthesis of new DNA strands during PCR. Later, we used the Hy3 restriction enzyme as molecular scissors. Because enzymes are shape-specific, Hy3 only recognizes and cuts a specific GGCC sequence. This allowed us to digest the DNA and eventually see the size differences on our gel. Unit 4 connects directly to the TAS2R38 receptor itself. This protein is a G-protein coupled receptor, or GPCR. When a bitter ligand, like PTC, binds to this receptor on the tongue, it initiates a signal transduction pathway. This cascade usually uses second messengers to relay the bitter signal from the cell surface to the brain. A single nucleotide polymorphism, or SNP, in the DNA changes the shape of this receptor. If the shape is slightly different, the ligand cannot bind. The message is never sent, and the person remains a non-taster.
Unit 5 focuses on heredity, which is how traits are passed from parent to offspring through genes. Each child inherits half of their genes from their mother and half from their father. Different versions of genes are called alleles, and some alleles are dominant over others. In the PTC tasting lab, the tasting allele is dominant, meaning tasters can have either one or two dominant alleles. Unit 6 focuses on gene expression and regulation. Regulatory sequences in DNA help control whether genes are turned on or off. The genes that are expressed in the cell determines the cell's function. In the PTC tasting trait, the TAS2R38 gene is transcribed and translated into a receptor protein. This receptor protein is found on the surface of the taste receptors and allows for some people to detect the bitter taste of PTC. Unit 7 focuses on natural selection and evolution. The TAS2R38 gene, which affects PTC tasting, has experienced relaxation and selection because environmental pressures for tasting bitter food in human have weakened. Mutations also create genetic variation within populations, giving natural selection traits to act on. Humans can also use artificial selection by choosing and increasing favored traits in populations over generations, such as the tasting trait. Unit 8 focuses on ecology and biodiversity. Greater genetic diversity in a population can increase resilience and survival. The TAS2R38 gene has multiple alleles which create both taster and non-taster phenotypes in humans. The diversity of TAS2R38 gene can also vary by location. Studies have shown that genetic diversity for this trait is especially high in many African populations.
Finally, we look at the bioethics of this technology through lens of Evangelium Vitae by Pope John Paul II. This encyclical emphasizes that human dignity is inherent and sacred. This raises a major concern. As we use PCR to screen for genes like BRCA1 or other health markers, do we risk creating a eugenic mindset where we value lives differently based on genetic perfection? While testing empowers us with life-saving data to protect the gift of life, we must ensure that a person's genotype, whether they are a taster or carry a high-risk mutation, never defines their worth as a human being.
In Laudato Si, Pope Francis explains that human life and dignity must always be respected. He warns that scientific research can become unethical when boundaries are crossed during experimentation on human beings. Every person has an unalienable worth, and protecting human dignity is more important than achieving scientific progress. Thanks for watching.